Okay, so now we're going to look at some anabolic pathways for metabolism. Okay, what am I doing here? So the anabolic pathways, anabolism is biosynthesis. This serves vital purposes in nutrient storage, nutrient synthesis, the structural element synthesis, and the synthesis of special molecules such as FAD and NAD, which are our electron carriers. Now, the body stores nutrients for two reasons. When nutrient intake from the diet exceeds the amount of energy immediately required, and when the body needs a ready supply of nutrients to oxidize for maintaining metabolic homeostasis between meals. Two main nutrient energy storage forms are glycogen, where we will store glucose as glycogen, and adipose, which is made up mostly of triglycerides. Now remember, we talked about the breakdown of glucose. So when we need it, we can break down glycogen to glucose and break down glucose. And then we talked about fats being broken down from triglycerides, being broken down by beta oxidation and glycerol. And we can store fats as adipose, or fat tissue. Now, glucose anabolism, we have two processes, glyconeogenesis and gluconeogenesis. These are two anabolic processes involving glucose. Glyconeogenesis is the storage of excess glucose obtained from the diet as glycogen. Glycogen is a large branched molecule composed of thousands of glucose units. It's a series of enzyme-catalyzed reactions, mostly in hepatocytes and skeletal muscle fibers, that add glucose units to a growing storage molecule. So essentially, you take glucose molecule and you form this very large starch of glycogen for storage. Gluconeogenesis, on the other hand, this is the synthesis of glucose molecules from non-carbohydrate sources. Once glycogen supplies have been exhausted, hepatocytes and specific kidney cells can convert 3 and 4 carbon compounds into glucose. Potential substrates could include glycerol, pyruvate, lactate, intermediate compounds from the citric acid cycle, and specific glucogenic amino acids. Fatty acids cannot be converted into new glucose molecules. Now, one of the reasons why we have gluconeogenesis is the brain needs to have a constant supply of glucose. And if you have no glucose sources, this is a way that the body, the liver in particular, can make glucose that can be used by the brain. Now, fatty acid anabolism, this is the synthesis of fatty acids, resembles beta oxidation in reverse. Although it involves completely different pathway, enzymes, and manufacturing location. This is a process referred to as lipogenesis. Genesis means formation. This is the process used to synthesize fatty acids. It takes place in the cytosol under the direction of fatty acid synthase, which catalyzes a reaction that progressively lengthens fatty acid chains to carbon units at a time. Remember, beta oxidation, you were breaking down 2 carbon units at a time. Most fatty acids are attached to glycerol and then assembled into triglycerides in the ER and stored in adipocytes. Majority of energy is stored as triglycerides in adipose tissue. Glycerol and fatty acids are derived from amino acids and glucose, not just from dietary sources. Excess glucose that is not stored as glycogen will be converted to an acetyl-CoA, and then that acetyl-CoA then can be stored as a fatty acid chain by going through lipogenesis. That's why if you have excess glucose in your blood that gets removed, it's going to be stored as fat. Glycerol can be synthesized from other products of glycolysis, and excess amino acids are then converted into triglycerides by a similar mechanism. We also have a disease referred to as fatty liver disease. The role of the liver in lipid metabolism becomes obvious in a condition referred to as fatty liver disease. Many causes, including alcohol abuse, obesity, diabetes, and many drugs and toxins, are characterized by the accumulation of triglycerides and other fats in the hepatocytes. As you can see here, normal liver versus a fatty liver. The exact mechanism in which fatty liver disease evolves isn't precisely known and may include a decreased capacity for mitochondrial beta oxidation, increased fatty acid synthesis, increased delivery of fatty acids to the liver, and a deficiency in the export of triglycerides from the liver to other cells. Untreated fatty liver disease can actually progress to cirrhosis and hepatic failure, generally reversible if the underlying cause is addressed. So if you actually are, say, obese, you can actually lose weight, and that will reduce your fatty liver. Treatments include dietary modifications with weight loss, abstinence from alcohol, and certain medications. Amino acids are not stored to any significant extent in the body's proteins. The body is capable of synthesizing 11 of the 20 amino acids. 9 amino acids must be attained from dietary sources. The synthesis involves reactions to add amino groups to carbon skeleton molecules such as alpha-ketoglutarate, pyruvate, and oxoacetate, which can all make amino acids, 11 amino acids. Cells synthesize limited number of proteins. Excess amino acids are converted to other molecules for storage. Your glucogenic amino acids are converted to glucose by gluconeogenesis and stored as glycogen. Other amino acids are converted to fatty acids and stored as fatty acid or adipose tissue. Okay, and that is it for the anabolic pathways. The final topic is going to be metabolic states and the regulation of feeding. So when we look at metabolic states, there are two primary metabolic states. We have the absorptive and post-absorptive states. The absorptive state occurs right after feeding. It's from the time the ingested nutrients enter the bloodstream and can last up to 4 hours. Following processes occur as nutrients are absorbed from the small intestines. Oxidation of nutrient molecules, primarily glucose, provides fuel to cells. Gluconeogenesis stores excess glucose in skeletal muscles and the hepatocytes. Lipogenesis stores triglycerides in adipocytes and hepatocytes. Protein synthesis provides structural material for cells, and the pancreatic hormone insulin orchestrates many absorptive state processes, and its release is triggered by an increase in blood glucose levels. So this is everything that sort of happens in the absorptive state, okay? And you just want to be aware of all the events that are occurring in the absorptive state, as this will be an open-ended question on your next exam. The post-absorptive state begins once nutrient absorption is complete. This is usually after a 4-hour absorptive window has ended. Although variable, the body is used in this state in late morning, late afternoon, and most of the night. Now, what happens in this particular state? Anabolic processes slow or stop, and following processes dominate. You'll have the breakdown of protein in muscle cells releases glucogenic amino acids into the blood. Ketogenesis in hepatocytes will convert fatty acids to ketone bodies and release them into the blood, which can be picked up by cells to undergo TCA cycle to produce energy. Gluconeogenesis and glycogenolysis in the hepatocytes release glucose into the blood. Lipolysis in the adipocytes releases fatty acids into the blood. And oxidation of molecules, fatty acids, provides most cells for the fuels. Glucose sparing, this is when cells catabolize newly delivered fatty acids preferentially to conserve glucose for cells of the nervous system. So glucose sparing is a process in which most cells in the body will actually preferentially use fatty acids to break down for energy so that the brain and other nervous tissue can have access to the glucose that is in the blood. Several hormones help the body adapt to the post-absorptive state. This includes glucagon, which is released when blood glucose levels drop and triggers glycogenolysis and gluconeogenesis in the liver. Epinephrine and norepinephrine stimulate lipolysis in adipose tissue and glycogenolysis in the skeletal muscles. Remember, epinephrine and norepinephrine are associated with your adrenaline rush, where you might need a lot of energy. Cortisol will stimulate gluconeogenesis and process that is going to release glucogenic precursors into the blood. And this looks at the post-absorptive state and everything that's happening there. So again, just make sure you understand what's happening with the absorptive and post-absorptive states. Fasting is a prolonged period during which little to no food is consumed. It may be intentional or it could be due to limited availability of nutrient-dense foods or to disease processes that prevents ingestion or processing of nutrients. One consequence of fasting is the condition referred to as protein wasting. Normally, after hours in the post-absorptive state, proteins are degraded and their amino acids are used as the primary source of carbon for gluconeogenesis. Remember, you got to keep the brain fed. Some degree of protein degeneration is normal and does not do harm to the body. However, this cannot go on for long periods of time as problems will begin to arise. The first noticeable in muscle tissue, where you'll have much higher protein content than other cells. The degradation of muscle proteins results in weakness and fatigue along with significant decrease in muscle mass. The body does not just rob muscles of protein. It does not discriminate among sources of protein or cell populations, and this can lead to loss of critical structural functional proteins, including proteins found in the heart, liver, brain, and in severe cases can prove to be very fatal. Now, feeding is controlled by a variety of hormonal and neural signals. This will stimulate or inhibit the feeding-related nuclei within the hypothalamus. The hypothalamus will house two nuclei that control the homeostatic variabilities that are associated with feeding. The satiety center, which elicits feelings of fullness and inhibits the desire to eat, and the hunger center, or the feeding center, elicits feelings of hunger and stimulates the desire to eat. On a more long-term look here, we have two hormones. Leptin, which is produced by adipocytes, stimulates satiety in the center and inhibits neurons in the hunger center. Ghrelin is a hormone that's produced by the stomach and the mucosal cells, stimulates neurons in the hunger center to promote hunger. The hunger center is also stimulated by a drop in leptin levels and allows the hunger center neurons to become activated. Short-term signals would include insulin, which is released in response to feeding. Similar actions of leptin decreases food intake. Feeding stretches the stomach walls, which initiates the release of gastrointestinal hormones such as CCK, which we talked about with the digestive system. Both stimulate vagus nerve to indirectly suppress the hunger center and decrease the release of hunger-related neurotransmitters, which would ultimately promote the feeling of hunger. And then finally, levels of certain molecules in the blood can stimulate or inhibit these hypothalamic centers. For example, low levels of glucose or hypoglycemia can stimulate hunger center and release some of those neurotransmitters which are going to promote hunger, such as orexin. And again, that's going to be it for the metabolism chapter here. Again, for my physics students, if you have any issues with any of these pathways, we could definitely hop on a Zoom call and I can take you through these things. My P4 students, this is probably not the first or last time that you'll be seeing these pathways, unfortunately. But even if you want me to go through these pathways in a little more in depth with you beyond the recordings, just feel free to reach out and we can, set up maybe a meeting time. We'll definitely talk about it in our Tuesday, Thursday sessions, but we can even talk about it a little bit if you want. Okay.